go ahead and just briefly talk our way through the basic stages of mitosis to make sure that's clear for everybody. If we pick up at G2 of interphase, the DNA has been duplicated, and you can see that on the left here, but it remains loosely packed. It hasn't started to condense yet, and it won't start to condense until M phase truly begins. The nuclear envelope's completely intact, and you notice we don't have a spindle apparatus yet, but there are a pair of centrosomes, and these centrosomes, also called microtubule organizing centers, are going to be responsible for building the mitotic spindle. So what I want you to pay attention to as we move through these phases, what's happening to the DNA, what's happening to the nuclear envelope, and what's happening to the spindle apparatus. So all the appropriate signals are received, the cell moves into M phase, and it begins with what the editors like to call prophase, where the mitotic spindle is beginning to form. What's actually happening early on here is that the centrosomes are this organizing center where microtubules are beginning to radiate outwards. These microtubules end up grabbing onto each other and pushing one another apart. So by growing and pushing apart, the centrosomes find their way to opposite ends of the cell and begin the process of elongating the cell a little bit. Nuclear envelope still relatively intact, and we see the beginnings of condensation of the chromatin itself and the sister chromatids becoming visible. So we would call this prophase. This is the very beginning. Now, your book divides up prophase into prophase and prometaphase. In prometaphase, what we see next is that the nuclear envelope begins to disintegrate into blebs, small vesicles of membrane that just sort of scatter throughout the cell, and they'll recoalesce later to form the two new nuclei. By now, the centrosomes are at opposite poles of the cell. The cells are probably elongated a little bit. There are microtubules that have found each other, and we call those non-kinetochore microtubules. And then there are some microtubules that, through random action, find kinetochore proteins in the centromeres. Now, kinetochore proteins play several roles. Their primary role is to attach to these microtubules and to follow along those microtubules towards the poles in anaphase on the next slide. So we've got kind of this tug of war that's going to start here in prometaphase, where the kinetochores get attached from both poles and start moving back and forth until eventually they all line up along the metaphase plate. When they do line up along the metaphase plate, what you see on the bottom left here is that they are all bound from both sides by the kinetochore microtubules. Once that happens, anaphase is allowed to proceed. The cohesins that are found in the centromeres are broken down, and the centromeres fall apart, and the two sister chromatids now become daughter chromosomes. They're not attached anymore, and they're headed towards separate cells, and so they're referred to as daughter chromosomes at this point in anaphase. Finally, once they're separated far enough in telophase, we see the blebs recoalescing and forming the nuclear envelope. We see the DNA beginning to relax and come uncondensed. We see the entire spindle apparatus beginning to depolymerize. Once all that has happened, finally, we can finish with cytokinesis. In animal cells, it's a cleavage furrow like you see here. And in plant cells, it's a cell plate. So my suggestion to you is to go through these different images, find some videos online if you need to, go through the figure that's in your book, go through these slides here, and make sure that you understand the flow of those three structures: the DNA, the nuclear envelope, and the spindle apparatus. And talk your way through, even do it out loud, what's happening to them as it moves from start to finish. Do that many times through before you start to label things as prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. The labels can come later. The first important thing is to make sure you understand the series of steps as things are moving from a G2 phase cell into two brand new baby cells in G1 of interphase.